The goal of this procedure is to obtain pure axonal preparations suitable for examination by conventional biochemical or immunocytochemical techniques. This is accomplished by first culturing embryonic dorsal root ganglion cells on culture inserts bearing a porous membrane filter. As the DRG neurons extend axons, some of the axons grow through pores in the filter and extend on its bottom surface, while the cell bodies remain isolated on the top surface. After the desired experimental manipulations, the axonal preparation is obtained by physically removing the cells on the top side of the filters while keeping the axons that grew on the bottom side. Ultimately, the isolated axons can be lysed for biochemical analyses or fixed for immunocytochemical examination.
By producing pure axonal samples suitable for analysis by conventional biochemical techniques, this procedure has proven to be very useful to investigate the physiology and pathophysiology of axons.
As you will see, the technique is relatively simple to implement and can be adapted to particular experimental settings. We are going to demonstrate its use in studying axonal degeneration mechanisms.
The day before plating, begin by coating the filters under a sterile tissue culture hood. First, place 24 mm filter inserts in a 6-well receiving plate. Then add 2 ml of 1 mg per milliliter poly-D-lysine in water to the bottom compartment and 1 ml to the top, and incubate the inserts for 1 hour at room temperature. Next, aspirate the poly-D-lysine.